What is the human microbiome, and why is it important to study it? The gut microbiome consists of bacterial cells living in the human gastrointestinal tract, which outnumbers the host cells by a factor of 10. Studying the human microbiome helps us understand how immune systems between hosts vary and how they develop according to lifestyle. Microbiome diversity is linked with good overall health, and we have options for new therapeutics, like fecal matter transplants. What is an organ on a chip, and what can it be used for? Organs on chips, or microphysiological systems, are cell or tissue cultures on microfluidic systems that can model organ function. Using organs on chips, we avoid having to use animals in experiments, we have more accurate analysis of tissue function, and we can connect different organ MPSs together to determine interactions between organs when studying a given disease. Why is it important to create a gut microbiome on a chip? What are the potential benefits and applications? Creating a gut microbiome on a chip would allow us to better understand how imbalances in gut microbiota are linked to diseases such as Clostridium difficile, inflammatory bowel disease, and type 2 diabetes. We can perform experiments without compromising ethical values associated with animal studies, and we do not need to radically change a patient's microbiome just for a study. We can also grow gut bacteria that are hard to culture, since current techniques limit the number of microorganisms that we can study in culture, and the behavior on a petri dish may not be the same as behavior in vivo. It can allow us to study interactions between gut microbiota and immune system in more detail, and give us controlled experiments where we can run a large number of trials using tissue from the same person. We can thus compare effects from changing microbiome diversity or introducing new bacteria to the system. The microbiome affects health and overall digestive system function, and growing human cells without the microbiota is unrepresentative of conditions in vivo. Integrating a gut on a chip with other tissues, such as the brain, would require microbiome, since microbiota have been implicated in other diseases like Parkinson's and obesity.